Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I'm Mr. Ulrich. In this video notes cast, I'm going to reintroduce the concept or the topics involved in biochemistry. And I'm also going to get into some water chemistry as well. It's important for us to, as AP students, be able to discuss on a structural level the biochemistry of the cell membrane and other structures within the cell and how they work. And so we're going to need to have a common language to do this. Science is, by its very nature, empirical. That means it tries to figure things out by breaking them up into smaller bits and then learning the properties of those bits and how those bits go together in order to create the properties of the whole. Now, when we start breaking larger compounds apart, molecules apart, we yield atoms. Each element, as we see here in the periodic table of elements, each element has its own characteristic number of protons. And that gives the element its properties. They're arranged this way in the periodic table. Mendeleev recognized that there is a pattern based on the number of protons. There's a pattern to the reactivity to the chemical properties that the different elements have. Now, phosphorus was first discovered by this guy, Hennig Brand. Hennig Brand, like many researchers of his day, was trying to figure out how to turn lead into gold. So he had his big idea. He had a great idea. His idea was to get a bucket and then pee in that bucket and save the pee. And he just saved the pee for a long time. And as the water evaporated from the urine, water contains phosphates, and through chemical degradation and natural breakdown of those materials that were in the urine, it started to glow. And that's where the name phosphorus comes from. Now, as biologists, we don't have to really concern ourselves with all of the elements here on the periodic table. There's just a few that we are mostly going to be talking about. The science of Einsteinium is not really pertinent to biochemistry so much, but carbon, nitrogen, oxygen, phosphorus, sulfur are certainly important. Hydrogen, ubiquitous, sodium, magnesium, potassium, and calcium. All of these are going to make up the vast majority of the compounds of life. It's not exclusive. Zinc is important, iron is important, selenium, there are plenty of other things that we need, but these are the major. When we start looking at the different elements and their characteristics, we see the pattern that was noticed by Mendeleev and others. It seems to correspond to the number of protons that were in each element. One of the major players here was not actually the number of protons, but the number of electrons that are whizzing around the outside, most notably the electrons that are in the outermost shell of the atom. If you remember back to chemistry, this is where the octet rule comes in. And we can arrange these atoms in columns based on the number of electrons they have in their outermost shell. We call those the valence electrons, and it does give them similar chemical properties, especially bonding, covalent bonding properties. The number of valence electrons that an atom has a direct impact on that atom's reactivity, on that element's reactivity, the number of bonds it's going to make and how quickly it makes those bonds. Because atoms want to have a full valence shell. Now, if that's the first valence shell, that's 2 atoms. If it's the second or third, there's going to be 8 electrons whizzing around there. So if they have a partially filled outer shell, it's only got 1 or 2 in the second or third shell, it's going to try to lose those. If it has almost full valence shell, then it's going to try to pick up a couple other ones. So you can see if there are atoms, there's one atom that really wants to lose an electron and another one that really wants to gain an electron. You put the two together and they can kind of switch it back and forth, switch it back and forth, switch it back and forth, switch it back and forth. And that sharing of the electron is what forms the covalent bond. So these two hydrogen atoms here are quite happy because they both have two electrons in their valence shell. Um, they're sharing those two, but sharing is caring. The properties of the atoms, the elements that make up larger biological molecules, are going to give us some of the information, some of the properties of those molecules. We also need to know how those are bonded together. Since most of the elements in living things are nonmetals, carbon, nitrogen, oxygen, phosphorus, sulfur, and hydrogen, they share electrons with one another to complete valence shells, and they form what are called covalent bonds. Now, there are other bonds that are important, like hydrogen bonds, but we're going to talk about those in the next slides to come. A couple more words on covalent bonds. They come in a couple varieties, depending on the nature of the atoms that that are sharing the electrons. They can either be polar covalent bonds or non-polar covalent bonds. And it all has to do, like I said, with the properties of the elements, how electronegative they are. If there is no difference in electronegativity, like between carbon and hydrogen, which have the same electronegativity, that means they share the electrons equally between the two, and there's no unequal distribution of charge. And that makes those molecules nonpolar. Also, because of that unequal distribution, because of the equal distribution of charge, it makes those compounds pretty stable. And stable compounds make for good building blocks of living things. They don't break apart. It takes a little bit of energy to make these compounds. That means that when the compounds are broken apart, they release that energy and have quite a bit of it.